Alright, this is an intro to Chapter 9, and we'll kind of get started here, and hopefully learn a little bit about the chemistry of the cell, a little bit about DNA. It's 9.1. Just remember to read the very first part of 9.1 and answer questions 1, 2, and 3 for your next class. So, a little background. When you think about everything that makes us who we are, or our hair color, eye color, all those things, it's all made possible by DNA, and it's coded by the 4 same chemical letters. We learned that in class to be, A, so let me do this, get this out of here a little bit, A, T, C, and G. And those 4 things allow us to be who we are. How is it possible? Just think of it this way. It's like letters in the alphabet. Where there's only 26 letters in the alphabet, there's an endless combination of words we can build from that. It's the same thing by just using these 4 letters in a variety of different ways. It's going to make a different code than if we used it something like this. Each one of those means something different, just like different letter combinations mean different words. And all of that together makes this image, which you can kind of look at and see are a bunch of people. All of those make the actual image of DNA itself. So where do we get this information? How did it come about? It comes about from the work. The first guy is a guy by the name of Frederick Griffith. And Frederick Griffith, back in 1928, he was attempting to prepare a vaccine, for pneumonia. He was preparing this vaccination for pneumonia, and what he did was he took a vaccine, and obviously what a vaccine is, it's just a substance prepared from an organism that's weak or weakened or possibly dead, and it's introduced into the body to help produce an immunity to it. We use this for, obviously you might have had the flu vaccine, there's a chickenpox vaccine, there's a lot of vaccines out there, but you take this weakened or organism that would not be able to survive, so a weakened microorganism, and you were to inject that into the body, and it aids in our immunity. What he did is Griffith worked with 2 strains of the bacteria. So he worked with 2 strains of the bacteria. The first type was the smooth type, and that smooth strain had a protective protein coat, and what it did is it caused the disease to be viral. In other words, that would cause the disease. If you had this, right, you wouldn't feel very good. The second kind lacked the protein coat, and we call that the rough. The rough lacked the protein coat and did not cause the disease. So that's his kind of basic experiment. Working with a vaccine, had 2 different strains of it. Let's see what he did. The first thing he did is Griffith injected a mixture of the heat-killed smooth bacteria and the living rough bacteria in the mice. Now, what he expected in this reaction is, if you got the heat-killed smooth and the living rough, the smooth bacteria and the rough, this one right here, it's dead. So it shouldn't cause any issue because you killed it off. This one's living, doesn't cause you to sick. He expected the mice to remain alive. Healthy. He expected them to be healthy. That's the block there. Because the rough cells do not cause the pneumonia. The smooth cells were dead. What actually happened was the mouse, they actually had pneumonia, and they actually died. They died of pneumonia. When Griffith checked the blood of the dead mice, he found that the living smooth cells formed. So all of a sudden these smooth cells formed. So just put this in kind of capsule. If you have smooth, so I'm going to represent smooth like that with a protective coat around there. You have that, you die. You have a regular, you live. You're able to defend it off. So what he did is he burned up this one. He was able to burn it up and injected that and inject the living one here. And what ended up happening was they still died of pneumonia. So what actually is going on here? Let's take a look at that. What he concluded was that somehow the dead cells were actually able to influence the rough cells to become smooth. He called this event bacterial transformation. We actually do a lab in my AP class where we take cells from a particular organism, in this case something that glows like coral or jellyfish, inject into bacteria. We take normal bacteria and we allow them to glow in the dark. Okay, we have them take the trait of another. So this kind of bacterial information is called transformation. You can actually see this kind of experiment here. So it explains a little bit better than I did. Example A, he takes the mouse, gives the smooth bacteria, it has that protective coat, mouse dies of pneumonia. If you have the rough, there's no protective coat, it'll live healthy. If you give it to the heat-killed bacteria, you killed the bacteria in there, the mouse would die, just like boiling your water kills off anything else. But if you were to mix these two together, this plus this, into there, what ends up happening is the mouse dies. It's because the heat-killed S mixed, and basically what ends up happening is this dead one gives a trait to that one. The trait allows it to grow a protective coat, therefore causing the death of the mouse. Something had to be transformed, had to be passed from this one to that one. So what is that transforming principle? Okay, so he said that smooth bacteria must be responsible. Griffith hypothesized that some chemical from the smooth must be responsible for transforming the traits of the rough into a disease-causing bacteria. What we find out the chemical is, in this case, the chemical was called, now that we know it, is called the transforming principle. Eventually it is known as DNA, but at the time he called it the transforming, sorry for the bad writing, I'm trying to do this with my finger, principle. And it was responsible for turning those rough cells into smooth cells. And those smooth cells were, in this case, lethal, caused the death. Took cells that would not be lethal and made them lethal. Later find out, now Griffin didn't know this, that it's going to be DNA, which is better known as deoxyribonucleic acid, that's responsible, DNA. That is later what's going to be named for the transforming principle. Okay, so it's the DNA that is responsible. So the last dot you have to fill again would be again transforming principle. Okay, so the next question is, after Griffith, we say, okay, well, what's this transforming principle? We know for a fact that the transforming principle is found on the chromosomes. If you remember back, Sutton was the one who told us that Sutton says that chromosomes are found in every cell, and in meiosis they're separated, and the transforming principle that Griffith calls it, the factors that Mendel called it, the genes, all that stuff is found on the chromosomes. So therefore, we look at the chromosomes and say, what are the chromosomes made out of? Well, they're made out of equal parts protein and DNA. Most scientists believe that one of the two had to be the genetic code, but which one is it? That's what we're going to look at next. Most scientists are going to actually think proteins are the genetic code. And we talked about this in class, the beginning class. Well, why? They do so much things. They're enzymes. They're structural. They do so much. They're more complex. They're rather large structures. So it's the fact that, and I apologize for the bad handwriting, that the heredity material was most likely proteins because they're more complex than the structure of DNA. And you can kind of see this here. Proteins are these rather large molecules. They can do a lot of different things. They're so complex. This is pretty simple. They have 2 letters that can be in the middle, and a total of A goes with T, C goes with G. Not very complex. So most scientists think this is the possible genetic code, and this is just something else. Not everybody, but most. All right, in 1944, okay, so right in kind of World War II wheelhouse, 1944, there was a guy by the name of Oswald Avery. And what he was able to do is through a very unique, very cool, simple experiment that DNA was responsible for the transformation. Okay, and I apologize for, I'm like a little child who runs out of room. I always remember that in grade school. I'd always keep writing and get to the edge. It's just a little hard with my fingers, so I apologize. Oswald Avery determines that DNA was the material, and I'll show you how. What he was able to do is he took a protein-destroying enzyme. Remember, enzymes can do catabolic reactions, and if you add them in the right place, they can tear things down. They aid an old chemical reaction. So he takes a protein-destroying enzyme, and he also takes a DNA-destroying enzyme. Okay, so those were his 2 different groups that he used. And he wanted to see if transformation, the thing that Griffith observed, would still occur. Okay, and so what he did was he found that transformation was prevented when the DNA-destroying enzyme was used, he found it was prevented. Okay, transformation was prevented. The passing on of the code was prevented when we destroyed the DNA. When we used just the protein-destroying enzyme, okay, it was not prevented. Okay, it was not prevented. And what this happened was, this was proof that in bacteria cells, the DNA code was the genetic code. Now, okay, this was affinitive because it was proof in bacteria, but not all cells. And what some people might say is, well, bacteria, they're simple. We're humans, complex. This is simple. Makes sense. Simple thing could be the code in bacteria. We're complex. Proteins might still be the answer. So all Avery did was able to prove it in bacteria, nothing more. The next experiment was going to be done by 2 people named Alfred Hershey and Martha Chase, and they did their experiment in 1952. And they were able to do that in 1952. What they did, and I'm going to type this up there, is they're going to use some dyes, and by using these dye, you can actually trace where that actually occurs. So what they did is they used a bacteriophage, and they were dyeing the different parts of the bacteriophage the different color. A bacteriophage is simply a virus that attacks bacteria, nothing more than that. And what it does is it has 2 different parts. So it's a virus, and it attacks and contains these 2 parts. Typically, you can represent a bacteriophage kind of like this. Kind of freaky looking. Not a bad drawing. Whereas the red part would be composed of proteins only, and the inner part is composed of DNA. Now think about this. If there's 2 different parts, and you radioactively tag each of the different parts, you could trace the experiment. And that's exactly what they did. And this picture is not very good. Let me see if I can change that. So here's kind of the experiment. What they did is, in this case over here, they took some radioactive P, because remember DNA has 3 parts: a phosphate group, a sugar group, and there's only phosphate in DNA. So they took some radioactive, in other words, it's just a dye they can follow, and they put it in the DNA. Proteins have sulfur, which DNA does not. So they put radioactive DNA in that so they could trace it. And then what they did was just followed it. Now they're labeled. Okay? They put them in a blender, they centrifuged, and what they found out was that the long story of this is the radioactive phosphorus was passed on to the progeny or the offspring from generation to generation to generation, whereas this was not. The sulfur was not passed on. It stayed with the original and died with it, confirming DNA is the genetic code because it's what's being passed on from generation to generation.
Hershey and Martha Chase confirmed DNA's preeminent role in genetics by demonstrating that DNA is the genetic material of a virus called phage T2. The phage, which infects E. coli, consists of a head, sheath, tail, and baseplate made of different proteins. DNA is packaged within the head of the virus. When T2 comes in contact with E. coli, the phage attaches to the bacterium by its tail. Next, the phage injects genetic material into the cell. The genetic material directs bacterial enzymes to produce viral offspring. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacterium. The bacterium breaks open, or lyses, and the phages are released. Since T2 consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components, so they designed an experiment to determine which it is. For their experiment, the scientists prepared T2 phages that either had radioactive DNA or radioactive proteins. To make the components radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells in two different culture media, one containing the radioactive isotope phosphorus-32 and the other containing the radioactive isotope sulfur-35. All the phages released from the bacteria grown in the phosphorus-32 medium had radioactively labeled DNA, while all the phages released from the bacteria grown in the sulfur-35 medium had radioactively labeled proteins. In the next phase of their experiment, Hershey and Chase infected E. coli with the two types of labeled T2. They then used a blender and a centrifuge to separate the protein parts of the phage, called the phage ghosts, from the rest of the infected cell. The scientists discovered that the labeled DNA appeared in the host cell, but not the phage ghosts, while the labeled proteins appeared in the phage ghosts, but not the host cell. In addition, the progeny of the T2 with labeled DNA were radioactive, while the progeny of the T2 with labeled proteins were not radioactive. Because genetic material is passed on from parent to offspring, Hershey and Chase concluded that DNA had to be the genetic material of T2. Their results helped convince the scientific community that DNA was the hereditary material.
Which gets us to our class notes. All of that work together combined over a 30-year period to tell us that DNA is the genetic code. And from that, we now know it is, and we're going to have to figure out the structure, which we figured out in class. Next time, we're going to look at how DNA replicates. If that's how it's built, how does it actually replicate? And that's what we're going to focus in on next time. Thanks for watching the video. Have a good day.